All right, let's wrap everything up with brain structure and function. So, starting out with the cerebral cortex. The cerebral cortex is kind of what you would think of when you picture the brain. It's that white, gray outer layer of our brain. So our brains kind of turn that color once we remove them, from our skull, and whenever we start to preserve them as specimens. My goal is to one day actually have a specimen that I can bring to class, but it's very difficult to obtain specimens nowadays. But that's a story for another time. We have two hemispheres, right and left. Let me get the pen out so I can write on the. There we go. What I want you to be aware of is we have a structure called the longitudinal fissure. It's that half down the middle where you can actually see the two halves of the brain. So we could actually stick our hands in there and separate the two halves. That's that longitudinal fissure. The corpus callosum is a thick band of nerve fibers that connects the two hemispheres together. So this thick band of nerve fibers right here. So it helps the two halves communicate together. And we'll talk about treatments for epilepsy later, but that's one of the treatments that we can actually have medications that slow down the rate at which the corpus callosum sends those electrical signals back and forth. And as we break it down further, we'll be talking about the four lobes in our brain. So first lobe is our frontal lobe. I've kind of color coded it for you so you can see. Our frontal lobe contains our prefrontal cortex, or the outer part we refer to as our prefrontal cortex. It has our human features such as self-awareness, personality, and executive functioning. It also contains our motor cortex, which initiates our voluntary movement. It also has probably, which you would think of it as around this area, it has our Broca's, not that area where I circled it the first time. It has our Broca's area, and it would be on our left side only of our frontal lobe, and it has the muscles that control speech. So if you're studying speak language pathology, you'll probably take lots of courses that study the brain and neurology, that the disorders that affect things like speaking, swallowing, etc. Whenever we hit our heads, or we have an injury to our head, or if you have a concussion, or if you ever hit your head playing sports, think about what's the first thing they ask you. They'll say, "What day is it? What's your name?" Because they're trying to see if you damaged your head, because that's something that would be damaged is your frontal lobe, your prefrontal cortex would be something to be damaged. Why would it be damaged? Well, think about your brain is in a closed system. Your skull is there's not much space between your skull and your brain. So if you were hit at a very high velocity, think about your physics that you took a long time ago. For every action, there's an equal but opposite greater reaction. So if you were hit from behind at a really high velocity, your brain would go jolting forward, and all that energy that sent your brain forward, all that inertia, etc., all that stuff, it would hit the front of your skull. That energy has to go back somewhere else, right? It's going to go shoot way back, and then it's going to do what? It's going to go back again. So even if you got hit once, your brain is going to be hit several times because your skull is a closed system. Your skull has nowhere to transfer that energy, so your brain will be hit repetitively. So that's why whenever someone hits their head in sports, or even think about a time if you've ever been in a near car accident, or if you've been in a car accident, if you've had whiplash, you lose your eyesight for a second because your brain slung to the back of your skull, and you hit your occipital lobe back there, which we'll talk about in a second. So your brain, very, very delicate thing. It's this squishy blob in our skull, and we think about our skull being able to protect our brain. Our skull actually has the potential to do more damage to our brain than we actually think about. Damage to your Broca's area can lead to a condition called Broca's aphasia. We can get Broca's aphasia by having a stroke. So if we reduce the flow of oxygen to Broca's area, we could damage those muscles that help us control that ability to produce speech. Our motor cortex initiates our voluntary movement. So if we're in an accident and we have damage to that motor cortex, we might have experienced difficulties with our ability to coordinate our muscle movements with our desired actions. We know that, well, we learned that our prefrontal cortex has a lot to do with our personality and our self-awareness because of Phineas Gage. In 1848, he was a railroad worker, and he had an occupational accident in which an iron rod was sent through his skull. So that's kind of a graphical representation of where the iron rod went through, and miraculously he survived. But if you look at the graphic, the reason he survived, which we'll talk about in a second, is the rod didn't pierce a vital part of his brain, the brainstem, but it did pierce his prefrontal cortex. And Phineas Gage, his family started saying that he wasn't behaving like himself. I read stories that he started gambling. He started just not being a very good person overall, very not like himself at all. So that was early on in 1848, where people that studied the brain, studied behavior, started to think that perhaps there were certain areas of the brain that were responsible for certain behaviors. And this was before even psychology was recognized as a science. So it kind of got people thinking about how the brain could affect behavior. Parietal lobe processes our body's information from the body's senses. It contains our somatosensory cortex. So this is where all of our body senses come in, and it travels out into our peripheral nerves and allows us to experience the senses: taste, touch, smell, all those things. The occipital lobe is responsible for our vision. So between the occipital lobe and our eyes, there's this big nerve called the optic nerve. It's really long. It travels from the occipital lobe to the back of our eyes. So that whiplash you experience if you've ever been in a car accident, that's that yanking from your eyeballs to the occipital lobe, is that yanking of that optic nerve. And chapter 5 has a lot more information about the eye, the anatomy of the eye, the different types of cells of the eye. And the eye is just, it's a really fascinating thing to read about that we just don't have the time to cover. But we learned that the eye has specific neurons that respond to certain stimuli. So we have neurons that respond to certain colors and shapes. There's cells that respond to circles. In fact, we have cells that we call grandmother cells that infants use to recognize their mom's faces. Infants recognize cells that are round, and we call them grandmother cells because they help us recognize round things. That's why we find babies cute. We also have those cells. We find round things cute, round puppies, round babies. That's why they hired psychologists to redesign SpongeBob 10 years ago. They added more circles on him because psychologically, we find circles to be visually appealing just because of the sensory stimuli in our eyes. So this is kind of just to show you where the sensory and motor cortex is. So that motor cortex is going to be in the frontal lobe. So it's going to be this one in the front. The sensory cortex is going to be in the parietal lobe. So it's going to be this blue one in the back. So it's kind of just the visual representation so you can see how they're split between the two different lobes of the brain. And the last of the 4 major lobes is your temporal lobe, and this is responsible for your hearing, memory, language. In your temporal lobe, I always forget the L. It sounds like I'm saying tempura, like vegetable tempura. It's temporal, but I'm saying, I almost sound like, because of my accent, I'm saying tempura. Temporal. We have Wernicke's area, which I kind of describe it as being like right around here. Wernicke's area is responsible for our ability to think and understand and interpret language. So if we ever have an injury to Wernicke's area, or we lose oxygen supply to Wernicke's area, we could experience Wernicke's aphasia, which would affect our ability to understand language. So, we would be able to hear, but we would not be able to interpret that language. So it's a problem that some people experience when they have injuries to Wernicke's area. So then we have subcortical structures, and there's a lot of subcortical structures. In this one, we're going to talk about some of them in more detail, but the corpus callosum we've already talked about. Your thalamus and hypothalamus and your pituitary gland are what we refer to as your endocrine glands. So these are the glands that kind of master your hormones. And they're also responsible for regulating some of your sensory information as well. Your brainstem we're going to talk about in a second, and then we have our cerebellum. Our cerebellum is going to be responsible for our balance and coordination. There's an old commercial that I think is kind of dated, but if I find the video, I'll post it on Blackboard. I think that would save us some time to talk about. But we'll be talking about the brainstem in a minute. So, like I mentioned earlier, when it comes to subcortical areas, with people that have epilepsy is caused by rapid communication between the two hemispheres of the brain. So we're having these electrical charges sent back and forth between the two hemispheres. So they came up with this procedure called the commissurotomy. They call it the split brain surgery, where they sever the corpus callosum, with the rationale being if that connection isn't there between the corpus callosum, the two hemispheres, then the two halves can't rapidly fire electrical charge between another. And people were able to reduce their seizures, but it came at a cost that affected memory problems later. Your thalamus is your sensory relay center, so that's kind of what I just want you to be aware of for your thalamus. Your hippocampus is responsible for learning and memory. I remember that because you come to campus to learn, hopefully not just memorize, but learn. Your hypothalamus, like I mentioned a minute ago, is linked to your pituitary gland. So these are things that affect your hormones and maintain homeostasis. So these are things like your drives for hunger, thirst, sex, and aggression. Your limbic system, we'll talk about. I don't think I have another slide for your limbic system. You want to be aware of your amygdala. So your amygdala is a structure in the brain that helps us process emotions like fear, and it also lets us process other emotions too. But it also lets us memorize things, and it has a very strong connection to memories tied to strong emotions. And it has a very direct root from our nose. So it has a very strong root from our sense of smell. So if you have memories that are triggered by smells, oftentimes those are directly a result from your amygdala. So if you've ever gone to like a funeral of a relative and you had family members that they were good 1 second, and they go and they smell like the cologne or perfume of their deceased loved one, and they just burst into tears, it's because that their amygdala can flood them with memories just based on that smell. And that's kind of what our limbic system can do. There's a video I'll show you later, but the amygdala is a really unique structure, and I like to talk about the amygdala. I'm just trying to keep this brief for the sake of time and keep it to the point. Let's see. So Henry Molaison, he's a case study. They refer to him as HM. He experienced seizures in 1953. They attempted to control his seizures by removing part of the hippocampus and amygdala, and they reduced his seizures, but he was unable to consolidate his memory. So he was not able to kind of put his memories together. But again, his seizures weren't as severe. So this happened in 1953. So back then, people were able to get away with some very, I would say, some very shady procedures. But I guess to his benefit, they did help him. But midbrain, do be aware of some of the midbrain structures. The tegmentum is what we would refer to as the VTA, the ventral tegmental area. And the reticular formation is a network of cells that help regulate our sleep-wake cycle. So the hindbrain, this is what I was talking about. So damage to our hindbrain, to these structures, your medulla is responsible for your autonomic processes, so your sleep-wake cycle, things you don't have to tell your body to do, your breathing. We don't have to tell our body to breathe. Our body does that by itself. So if our medulla gets damaged, our pons, which connects our brain to our spinal cord, and remember, we have a central nervous system that relays all the information from our brain to the rest of our body. So if that gets damaged, oftentimes it's not good. The outcomes are not very good. Cerebellum sends out the messages from our muscles and tendons to the joints. So damage to the hindbrain structures often, this is where we either are comatose or we die. So people that often have injuries to the brain can sometimes survive if the brain stem was not seriously hurt, or where the oxygen supply was not reduced to the brain stem. So studying the brain a long time ago, it used to be done with wet specimens. So somebody dies, we ask them if we can have their brain. We cut the brain out, we slice it, and then we run tests on it. We look at it in the microscope. Nowadays, it's done with more modern technologies. So we have a CT scan. CT stands for computerized tomography scan. So X-rays pass through the body tissue, and we can see tumors, growth, shrinkage. So this one is a CT scan. You can see a tumor. And this is a really neat one because you can see the corpus callosum, you can see the longitudinal fissure. Let me see if I can get the pointer. You can see the longitudinal fissure, you can see the corpus callosum, you can see the cerebellum right here, but there's this big tumor that has eaten up part of the frontal lobe. So this person is likely experiencing some behavioral issues or some personality issues, some executive functioning issues. We can't say that that's causing it, but we can definitely say there's going to be some causal, some correlational things there by the size of this tumor. We can almost possibly think that it could be cutting into the motor cortex just by how big the tumor is. We have MRI. MRI stands for magnetic resonance imaging. So a strong magnetic field is generated to move hydrogen atoms. Hydrogen atoms emit electromagnetic radiation. So you can kind of see that this tumor was highlighted by those hydrogen atoms emitting that electromagnetic field. So this tumor looks like it's in that parietal lobe, and it could be in that somatosensory cortex. So this person could be experiencing a whole slew of issues. It could even be kind of digging into that temporal lobe. So you can see that we're able to find tumors and pretty much any irregular abnormality. Tumors aren't the only thing that can be affecting the brain. Sometimes areas of the brain can get damaged from blunt trauma, from lack of oxygen, lack of glucose, and there's all kinds of things that could cause the brain to sustain injuries. PET scan. PET stands for positron emission tomography. So we inject the brain with a mildly radioactive glucose. The reason we use glucose is because glucose is your brain's energy source. Glucose is your brain snack of choice. And so this is why people that do keto diets, they get headaches and stuff, because whenever your brain is deprived of glucose, your brain will produce ketones, and ketones are not as sustainable as glucose. So your brain will produce ketones in an effort to try to sustain itself to get you through the day. And your brain produces ketones to try to get you to get glucose because your brain really wants glucose. And it costs your brain energy to make ketones. And your brain, like I've been telling you, is lazy. It does not like to use energy. So the mildly radioactive glucose, we can stick it into our body, and we can actually trace its movement, and we can actually see where it goes because of the reactivity. So in resting state, we can see that the frontal lobe is active because, again, that's where our awareness is, our personality is. And we can see that the cerebellum brainstem is active. During language and music, we see that temporal lobe is active, Wernicke's area, Broca's area, cerebellum frontal lobe. Now, interesting thing is whenever just language and music is active, you see that only Broca's area and then only Wernicke's area is active. So be aware of which side Wernicke's area is on and which side Broca's area is on. fMRI stands for functional magnetic resonance imaging. It has the same principles of MRI, but it allows us to see the function. I like this picture a lot because you can see the brainstem, the pons, the medulla. You can see the cerebellum. You can see the corpus callosum. And you can really see the folds and grooves of the cortex. And that is our crash course in brain structure and function.